Hello, everyone, and thank you for joining us this evening for the first week of the Rugby Research and Injury Prevention Group 2026 Journal Club. We will begin our first Journal Club presentation with myself. My name is Danielle Jefferson, and I will be presenting on sleep disturbances in elite athletes, psychological mechanisms, performance implications, and evidence-based interventions. This article takes a comprehensive look at how sleep is a significant performance and health issue for athletes at the elite level. So, why does sleep matter so much for athletes? Let's really start with the numbers that this article covers. Research shows that between 50% and 70% of elite athletes experience sleep disturbances. This number gets even higher at 70% for athletes that are competing in individual sports such as swimming or gymnastics, compared to the 58% for athletes in team sports. For context, this is not just one instance of a sleep disturbance. This is chronic, reoccurring disruption driven by a very specific set of stressors that elite athletes face. High training volumes raise cortisol and adrenaline, which physiologically delay sleep onset. Once you add to this the psychological pressure of actual competition, fear of injury, and constant travel across the time zones, other risk factors, you create conditions that are almost perfectly designed to disturb a good night's sleep. And what makes this particularly difficult is that it creates a negative feedback loop, which raises cortisol, disrupts sleep, and further raises the stress for the athletes. The authors of this article cited this as a vicious cycle, and breaking it requires targeted intervention. So, the authors had four main goals with this review. Firstly, they wanted to pull together all of the current data on how widespread sleep disturbances are affecting athletes currently and what causes them. Second, they wanted to really explain and identify the psychological and neurobiological mechanisms underneath why athletes are sleeping badly, and specifically at the brain level. Third, they wanted to quantify the performance and health consequences, asking what exactly happens to an athlete who is chronically sleep deprived. And finally, they evaluated a range of evidence-based interventions to see what actually works to improve sleep in athletes. Their findings included CBT, mindfulness programs, sleep hygiene education, and biofeedback, which we'll cover a bit later. So this review was conducted as a narrative review of literature published between 2013 and 2024. The authors drew from three primary fields: somnology, which is the scientific study of sleep, sports psychology, and psychiatry. The sources they analyzed all included meta-analysis, randomized control trials, and neuroimaging studies. They gave particular attention to the relationship between sleep, stress, and cognitive function in the elite athletes. In terms of actual interventions, they evaluated 5 different approaches: cognitive behavioral therapy for insomnia, or CBTI, mindfulness-based stress reduction, known as MBSR, personalized sleep hygiene education for the athletes, and biofeedback and neurofeedback, as well as multidisciplinary collaboration between sports medicine, psychology, nutrition, and various coaching staff. One other important thing to note is that this is a narrative review and not a systematic one, with a formal PRISMA protocol, which will really impacts the limitations of this review, which we will acknowledge later. So, the key findings of this review, I believe there are 4 real statistics that I want to focus on, as I think that they're most impactful, especially with the work that RRIPG does. Firstly, 50 to 70% of elite athletes are affected by sleep disturbances. This is a majority of elite athletes, which highlights a real problem. Second, 1.7 times higher injury risk for chronic sleep deprivation in athletes. Athletes who get regularly fewer than 8 hours of sleep are 1.7 more times likely to sustain an injury. This statistic is specifically from Helson's 2023 meta-analysis, which is cited in this review, and it's clinically significant. Third, just 3 consecutive nights of sleeping 5 hours or less can significantly reduce sprint performance by 3.1% and increase perceived exertion by 17%. In a competition, this means a 3% performance drop. can really be the difference between winning or losing, significant when you are looking at the performance of athletes. And finally, a 70 to 80% is the efficacy rate for CBTI in athlete populations, meaning that it outperforms medication and has no side effects. I do also want to touch base on the neuroscience side, as neuroimaging shows that sleep deprivation reduces an activation in the prefrontal cortex. And for an athlete, this means that they can lose executive functioning mid-game or mid-race, and that can be catastrophic to the athlete's actual performance. So, talking more about what these findings mean, this discussion really brings up a critical reframing of how sleep disturbances in athletes are not a byproduct of hard training, but they're deeply intertwined with psychological states and things such as pre-competition anxiety, cognitive hyperarousal, and emotional regulation deficits, which can all have a significant impact on the athlete's performance. What this means more practically is that we cannot fix the sleep problem by just telling an athlete to, go to bed earlier. We really have to look at things like sleep hygiene and psychological drivers. This can be rumination about performance, fear of failure, self-critical thinking, and other things regarding mental health. These things are also active at night, and it really impacts the amount of time that it can take for an athlete to fall asleep. It needs to be addressed directly. Another important point that the authors emphasize is that no two athletes should be and can be looked at identically. For different sports, different schedules, and different personal psychological profiles, they all have individualized demand and therefore require individualized approaches. For the treatment side of things, authors are also clear that CBTI is the gold standard, as it directly targets the maladaptive benefits about sleep and uses things like stimulus control and sleep restriction to rebuild healthy pattern, actually targeting the issues that are affecting these athletes. And finally, just with multidisciplinary collaboration, the stress for just the need for multidisciplinary care between sports medicine, nutrition, coaching, and psychology all really come back to sleep. No one single thing can address it alone. So if we want to see our progress in these athletes, sleep is the next thing to look at. I do want to cover the limitations of this review, as no study is without limitations. The biggest methodological issue for this review is that it's a narrative review, which means that the authors had discretion over which studies to include and how to interpret them, which introduces the possibility of things like selection bias when we're looking at our data. There's also a noticeable reliance on Russian language theoretical frameworks, such as excuse my mispronunciation if I do, Kasakatkin, Kovalazan, and Yakupov, which are the authors that they use alongside Western clinical literature. These perspectives in the actual review were interesting, however, they don't always map and transfer over neatly into the evidence-based frameworks that we typically see and use in sports science. Some of the other cited empirical studies for this review are referenced without deep methodological critique, which I also thought was interesting. This affects the effect sizes and confidence intervals aren't consistently reported, which also makes it harder to judge the magnitude of these findings. Finally, because this is a review and therefore does not have original data, we can observe these associations, but we cannot establish causality. We know that poor sleep correlates with poor performance, but the directionality and mechanisms still need further longitudinal study. For key takeaways, sleep really is a performance tool, and we want it to be treated as such with the same intentionality as training load, nutrition, and recovery for these athletes. Especially because it measurably raises injury risk, which of course is what we focus on. As for athletes, they should be establishing a consistent pre-competition sleep routines and wind down protocols so that when these problems do persist, something like getting a referral to a sports psychologist or someone trained in CBTI can be far more effective than relying on more conventional like sleep aid mechanisms. Righty. I also didn't have it on the slide, but I do want to touch on for sports organizations. For sports organizations, that the recommendations in this review they were to implement routine sleep screening as a part of almost every athlete's medical assessment, and as well as train sports psychologists in CBTI, MBSR, and specifically biofeedback. Building team-specific sleep protocols that can account for travel schedules and competition calendars were also cited as a recommendation for specifically sports organizations. That pretty much covers the study. I will leave the references on here just briefly for anybody who wants to follow up with this source. Dr. Lopez, do you have anything to add? Ooh. Dr. Lopez, you are muted.
Thanks so much for that. More importantly, did you go back 2 slides, or was it 1 slide where they, not your cover slide?
One more?
No. So for sports organizations, 1 slide, Danielle. Thank you. For sports organizations, they're telling you, oh, for all sleep screening. Remember, right? If you're going to go into sports, 90%, 95, maybe 97% of your sport cohort will be community sport. I do not think that they're going to be sleep screening. Natalia, did they sleep screen you when you were in soccer?
I didn't play soccer.
All right, what was the sport you played that you got?
Gymnastics?
Gymnastics. Did they
No,
go over sleep screening with you?
no.
All right. Don't correct me. Just kind of roll with me with what I'm trying to tell you. Right? So, no, they didn't do that. So, obviously, and Natalia, you played a pretty high level. And then at rowing, are they sleep screening you in rowing?
No, not at all.
So, as I was saying, she's at university level and she was in a very high level club gymnastics. So, basically, you have athletes who have played, even myself, semi-professionally in 22 countries, and they do not screen you. So this is a great study. It's ideal. What would you propose, Danielle? Now that you've got this knowledge base, what would you do at a community level if you were taking care of a girls' club team?
If I was taking care of a girls' club team, I would likely, my first default would likely be with a training sports psychologist in evidence-based feedback interventions.
You're
This
gonna
way,
wait a second. You're gonna find a you're gonna find a girls' soccer team that's gonna pay for a sports psychologist.
no.
Like I said. So let's be realistic, right? You have to kind of almost come up as and you will all be this, you will all do this, right? Liliana's dad did this. You will all be coaching your children at one point or another. So you have to do the best you can, and you have to take this information and you have to kind of whittle it down where it's kind of, hey, don't eat so late. Make sure you're getting rest post match days or whatever court days or game days. So this way they understand that. You have to kind of almost, every time you look at these studies, everyone, you almost have to really anticipate how will you implement this in your population. Or let's say, Dr. Cooper there, she's going to be a doctor in physical therapy, and she's going to be dealing with the pickleball 80-year-old crowd. So you need to tell them, hey, we do understand as a octogenarian, you won't need a certain amount of sleep. But if you're going to play pickleball, you may need to rest more. You may need to incorporate naps in your day. So you have to adapt. Does everyone understand what I'm saying? To who your team is, right? Whether or not your team is going to be, the pickleball squad, or it's going to be girls, your daughter's club squad, whatever it's going to be, you have to be that person. And again, they do these great studies under these circumstances, you know, uh, and again, you did say there was a limitation of a narrative. So they could take this information and, it sounds great and all, but it's very impractical. That's why a clinical study would be better to really evaluate this. But anyway, I'm
Gotcha.
just trying to make sure everyone understands, right? You're not going to be able to get the Girl Scouts to pay for a team psychologist, nor, you got to kind of work with what you're given. And I'm going to tell you, they're going to give you even less. So you have to really kind of try to do the best you can. Okay? And it will be a continual tug of war between you and the coach. If you all are interested in healthcare professionals in some capacity. All right? Thank you. Anything else on that, Miss Danielle?
That is all. Thank you for the feedback, Dr. Lopez. Now we can move on to our next presenter. Our second intern that will be presenting is Jordan Hall, and I will pass it along to you. Let me know if you have any issues screen sharing.
Hello, everyone. I'm Jordan Hall, a junior at Kent Place High School in New Jersey. Today, I'm going to talk about a study published in the journal ACS Sensors called an ultrastable wearable electrochemical sensor for prolonged monitoring of electrolyte loss and hydration. Wearable tech is great for counting steps or tracking heart rate, but what this article is really about is chemical tracking, and if you don't know what that is, it's actually measuring what's inside of our bodies through tracking sweat. Today, I'm going to be showing how this research team built a wearable skin patch that solves one of the biggest engineering problems of this field, making everyday health tracking much more reliable than it has previously been. To understand why the researchers wanted to create this wearable patch, it is important to start off with the background. Staying hydrated and keeping athletes' electrolyte levels balanced is imperative for the human body, as over 60% of our bodies are water. If you lose too much sodium or water, your physical and mental performance plummets, and it can even lead to dangerous medical issues. Because sweat is packed with information about our hydration and athletes' hydration, like sodium and potassium, which are two main sweat electrolytes, it is the perfect fluid to track in athletes' bodies. Collecting it is also completely non-invasive, meaning that there are no needles required. Herein lies the problem with current sweat sensors. They work great in the lab, but when it's tested on moving active athletes, they usually fail. When an athlete starts running or working out, two things can happen. First, their skin can rub against the sensor, and second, a heavy rush of sweat can constantly wash over the sensor. This causes what we call a sensor drift and what they referenced as a sensor drift. And this essentially means that the gadget's readings start drifting and become inaccurate. Because of this flaw, current sensors and previous sensors usually require tedious calibration. And if you don't know what that is, it is making a precise measurement right before you use them, and they don't last very long for athletes. Most models aren't ready for the real world and can't accurately help athletes monitor their hydration and electrolyte intake. Which leads me to the purpose of the study. The researchers wanted to take this technology out of the lab and make it practical for everyday consumers, athletes, and workers. Their goal was to create a tough, long-lasting wearable patch, and they named their invention the R sensor. The main objective was to build a device that can measure both electrolyte levels and how fast you're sweating at the exact same time over hours of heavy exercise without needing to calibrate like previous models needed. So there are two forms of the methods. First is how it's made, and second is the testing. So how did they do it? To fix the issue of the inaccurate readings caused by rushing sweat, the team used a special semi-solid electrolyte gel. This is a jelly-like material that combines the structural security of a solid with the flow of a liquid, and they mixed it with a water-repelling layer right inside of the wearable sensor. The water-repelling layer acted like a shield, and it kept the sweat flowing smoothly over the sensor rather than letting in a messy film and having water get trapped underneath the patch. This trick was what made the device calibration free because all you had to do then was stick on the wearable sensor without worrying about any precise measurements or calibration. Additionally, they needed a way to measure exactly how much the athlete is sweating. Instead of just guessing based off of skin moisture like older models, they built tiny microchannels that forced sweats into individual droplets. As these droplets passed by, the sensor counted them one by one, and it gave an incredibly accurate measure of the actual sweat volume of the athletes. The second method was the testing. So, in order to prove the patch actually worked, they moved to human testing. They put the R sensor patch on volunteers and had them do intense workouts. This included indoor stationary cycling sessions where conditions were controlled, as well as outdoor running trials where the weather and movement were unpredictable. The patch sent all the data wirelessly to a smartphone app so the researchers could track the volunteers' hydration level changes in real time. This leads me to the result of the study. So after doing all of this study or testing, they ultimately got to the results of the study. The R sensor performed beautifully. They saw first that the electrolyte readings were highly sensitive and incredibly stable. Even with the hours and hours of sweat washing over the patch, the readings did not drift or glitch. And second, the droplet counter perfectly tracked how fast people were sweating, spiking just at the right time when the volunteers pushed harder during their workouts. And finally, it proved to be truly ready to use because when the water repelling design, the sensors didn't need any prep work. They gave accurate numbers the moment the workout started, and right from the get-go, they were able to tell the speed of the sweat and how much the athlete was sweating. So moving on to some data, specifically regarding how long the device lasts. If you look to the top right in figure S4, I'm going to be talking about that for a little bit. One of the biggest accomplishments of the study was the sensor's shelf life. The team left the patches in storage and tested them over time. They found that the R sensor could sit on a shelf for over a month, and in this photo we see 2 months. And they found the R sensor and they found it would still work perfectly on athletes without losing its accuracy. Along with this, during the live workouts, the patches data matched up almost perfectly with the high-tech, expensive laboratory equipment, as we can see in the lower chart that I have provided on the slideshow. And the data proved that the sensor's internal setup effectively locked its accuracy in place, resisting both body sweat and vigorous physical movement. This leads me to the discussions of why does this matter for the future? The authors explained why this matters for the future by emphasizing that by getting rid of the need for manual calibration, they have turned a complex laboratory tool into a simple tool that can be used by hundreds, if not thousands, of athletes. They also discussed how tracking both salt concentration and sweat volume made major changes to their own research and research for futuristic goals. They found that if the sensor tells you that you are losing sodium, that's only half of what's actually happening. However, if it tells you exactly how many milliliters of fluid you've lost, an app can calculate the exact amount of sports drink you need to consume to recover safely. It makes hydration advice completely personalized to the athlete wearing it and makes electrolyte intake information much, much easier to obtain. There were a few limitations. There were a few limitations in the study, but they were still there. First, the human tests were done on a small group of healthy volunteers. To make this a mass market product, it needs to be tested on a much larger and much more diverse group of people. Second, the sweat changes depending on the weather and skin temperature of an athlete. Right now, adding in a built-in temperature sensor would make the math even more accurate than it previously was. And third, while it tracks sodium and potassium perfectly, manufacturing this patch to track other things like glucose or stress hormones, which they talked about in their research paper, is going to take a lot more engineering and a lot more time, but is very much necessary. This leads me to the key takeaways of the study, which first I want to talk about how the R sensor fixes the classic sensor drift problem that was in the previous models by using the clever water repelling design. Second, it was truly ready to use and last for over a month in storage. We saw the 2-month data. And third, by tracking both salt loss and sweat volume together, it gives us a real-world tool for smart personalized hydration. If you want to read the full paper or look at the exact charts, here's the reference to the study published in ACS Sensors. Thank you so much for your time.
Dr. Lopez, do you have anything to add before we?
Yeah, if Ms. Hall, bring us to the last, I think it's your last slide, the limitations. Go back.
Yes.
Yeah, did sweat composition vary between temperature and weather conditions? Are you telling me the actual results or outcomes from the unit itself gave you variations?
No, but this is something that I found that they hadn't tested just yet. They hadn't found this in their study yet. And I think it's important to factor in that sweat composition varies.
Okay, good. I want to make sure that was clear to everyone.
Yes.
Right?
Sorry if that wasn't clear.
The unit itself wasn't tested.
Yes.
Am I correct on this? This is what you're proposing that they need to take into consideration going
Yes.
forward, right? I mean,
Precisely.
everyone knows this, right? Everyone has played sport or been outside watching it. The humidity, the weather conditions, whether it's raining. I don't know how many sports I've played in the rain, but I would assume that there's a certain amount of dilution
Yes.
for the topical surface sensors that they got to be careful with that, right? I mean,
Yes.
and again, everyone needs to take these studies with a grain of salt, that they understand that most of these studies are done in like perfect ideal windows, so they may have maybe did they discuss because I know that they did try doing something like this with a group of athletes, and they used like saran wrap to try to isolate it. Did they discuss any of that? Any
No, they didn't
barriers?
talk about they didn't do any testing on temperature. They did it in a very controlled environment, which I thought was a huge limitation because how is that supposed to give real world examples if most of their healthy volunteers were in a controlled environment?
Okay. Everyone understands, right? When these surfaces or these surface sensors are on the on the skin, whether it's sweat, humidity, rain, uh, you know, I, I don't even know how they would conduct this in Hong Kong. Um, we could barely breathe. The humidity was so thick.
Hmm.
So I don't know, like in some areas, like on the planet, I don't know how that would be applied. I mean, don't get me wrong, there's tons of opportunity for them trying to get this sensor out. I think, obviously both these topics are really interesting. And not only that, but we all understand loss depletion, right? Um, the supplemental drinks wouldn't be doing so well financially if they didn't have a certain amount of impact, if everyone understands what I'm saying, right? Obviously they're well aware of the loss, but quantifying it is really the challenge overall in that. All right, next. Thank you so much. That was good.
Thank you so much, Jordan, for that wonderful presentation. Our third intern is Elon Cooper, and she will be presenting on traumatic brain injury and risk of neurodegenerative disorder. Elon, you're more than welcome to screen share. and take it away.
All right, can you all see my screen? Okay. Good evening, everyone. My name is Alon Cooper. I'm a rising senior kinesiology major at Hampton University. And today I will be presenting on Dr. Benjamin Brett's and his colleagues' review on traumatic brain injury and risk of neurodegenerative disorder. Okay. This paper brings together evidence from clinical and imaging studies, large cohorts, and post-mortem neuropathology to ask the question, does TBI increase long-term risk for neurodegenerative diseases like dementia, Alzheimer's, and Parkinson's disease? Before getting into the paper, I want to define a couple of terms. TBI stands for traumatic brain injury due to external force and classified by 3 levels of severity, mild, moderate, and severe. Neurodegeneration means gradual loss of brain cell structure or function as time progresses and can eventually affect thinking, memory, or movement. The major neurodegenerative disorders discussed in this paper are all-cause dementia, Alzheimer's disease, Parkinson's disease, ALS, frontotemporal dementia, and chronic traumatic encephalopathy, or CTE. One especially important term is polypathology, which means multiple disease processes can appear in a subject's brain at the same time. This idea matters because TBI-related outcomes are often mixed rather than cleanly tied to one diagnosis. And the last thing I want to define is amyloid beta and tau, which are proteins that are essential to brain function and after injury are linked to neurodegenerative diseases. This article is a narrative review, so the authors are synthesizing many different types of studies rather than reporting from one data set. A major takeaway from the background literature is that TBI of any severity has been associated with a 63% to 96% increased risk of all dementia. But the picture gets less clear when researchers try to link TBI to one specific disease like Alzheimer's alone. The evidence linking TBI is stronger for general dementia risk and Parkinson's disease rather than especially after moderate to severe TBI. One reason the literature is complicated is the studies define TBI severity differently, measure outcomes differently, and follow patients for different lengths of time. The last part of the paper focuses on how TBI might biologically lead to later neurodegeneration. The authors organized this around 3 major pathways, which are axonal injury, abnormal protein buildup, and chronic inflammation. Axonal injury is where the injury disrupts transport inside neurons and can lead to white matter degeneration over time. Abnormal protein biology, especially amyloid beta and hyperphosphorylated tau, may accumulate or become abnormal after injury, potentially contributing to plaques and tangles. Lastly is chronic inflammation, and cells like microglia and astrocytes may help immediately after injury, but if that response stays for too long, they can start contributing to long-term damage instead of repair. The important idea is that these pathways most likely work together and not in isolation. The purpose of this review was presented in threefold. They examined the progression of TBI and its association to different neurodegenerative conditions. They compared findings from different methods of study from clinical diagnosis to post-mortem studies, since clinical studies can provide long-term evidence while post-mortem studies only show a singular snapshot of the subject. And the last fold was to explain how acute injury can set off processes that continue for years and possibly contribute to neurodegeneration. Since this article is a collection of previously published studies, each study used a different method of collecting data. Some of the methods are large administrative and population-based cohort studies, post-mortem neuropathological work, meta-analysis, and PET imaging scans. From the research presented, authors were able to focus on different disease risk patterns and biological mechanisms associated with TBI and neurodegenerative diseases. Post-mortem studies often show that polypathology, meaning it's not only one isolated disease. Most large studies link TBI to a larger risk of all-cause dementia. One study that was based on World War II veterans reported that moderate and severe TBI were associated with 2.3 to 4.5 times higher Alzheimer's risk but other large investigations have not consistently reproduced that evidence. Parkinson's disease also appears more consistently associated with TBI rather than Alzheimer's disease does, but they're looking to increase research in that field. The largest scale study on former professional American football players were able to find that they have an increased risk of neurodegenerative deaths compared to the general population, but studies linking these subjects back to specific neurodegenerative diseases have come back inconclusive. In the discussion, the authors argue that TBI severity matters. Most severe injuries create clear structural disruption, and that seems to translate into clear long-term risk. They also emphasize that TBI does not lead to neurodegeneration through one single disease pathway. Instead, it may trigger a combination of white matter damage, protein abnormalities, vascular changes, and chronic inflammation. This helps explain why the clinical variety is seen across the studies and why postmortem brains often show polypathology instead of one clean pathology pattern. The broader implication is that future studies will likely be more informative if they separate severe injury severity and use biomarkers that track specific mechanisms. The limitations in this study are more about the state of the field rather than the review itself. The studies vary a lot in how they define TBI, how they diagnose dementia, and how long they follow the participants. Administrative cohorts are useful for large sample sizes, but they can often miss people that never sought out care or were diagnosed inaccurately. Broad severity categories also blur the differences between mild, moderate, and severe and repetitive injuries. Postmortem work is complicated by polypathology and by other contributors like aging, genetics, lifestyle, and vascular disease. This study shows a summary of significant research identifying TBI as a risk factor for developing neurodegenerative disease later in life, while also recognizing the gaps in research and the obstacles that prevent finding strong evidence to link TBI to neurodegenerative diseases. The biggest takeaway is that TBI should be considered an important long-term risk factor for neurodegenerative diseases, especially all-cause dementia and in Parkinson's disease. The evidence is strongest for moderate to severe injuries, while evidence for specific diagnoses like Alzheimer's disease is more variable. The biology also appears multifactorial, including axonal injury, abnormal protein handling, vascular effects, and chronic inflammation. The future of this field depends on better biomarkers and better designed longitudinal studies that separate injury severity and track progression over time. This is the reference of the article that I use in case you guys would like to go read more about it. Are there any questions or anything to add?
No, thank you. Oh, am I muted? No, I'm not. Okay, thank you so much, Elon. That was a really good review topic, and not only that paper, just to introduce everyone to the subject matter. I think when they're talking about it, I think not too long ago we came across a paper that they were trying to push the envelopes of what biomarkers, and I'll have to go back into it. There was a paper that they were trying a new biomarker, but again, it was the sensitivity was not that good, so it was really kind of criticized on their use of it. But again, I'll get that paper once I look back in my reviews. But again, I think that was a good review overall, everyone, to get an idea of what the subject matter and the impact of it is. Obviously, they should be considering TBI as a major risk. You know, it shouldn't be excluded within the possible risk factors for developing long-term neurodegenerative disorders or neurodegenerative signs and symptoms of aging, whether it was from the theater of warfare or sport. You know, you got to consider both of those have impacts, and they both affect the athlete or the personnel the same. And even if you had maybe, you know, I don't know if you realize it, but, you know, they say automobile accidents is number one cause of traumatic brain injury. You know, so make sure you kind of even consider that when you're evaluating athletes or evaluating patients. You want to consider that. That's also a potential risk factor. All right, everyone. Great job there, Elon.